Good evening, everybody. I'm Alistair Burt. I'm the Executive Dean of the Faculty of Health and Medical Sciences. I'd like to start by acknowledging the Ghana people, the original custodians of the Adelaide Plains and the land on which the University of Adelaide's campuses here at North Terrace and at Waite and Rho-GTPase are built. It gives me great pleasure to welcome you this evening to the biannual Abbie Memorial Lecture. Thank you for joining us. The Abbie Memorial Lecture, held in memory of Professor Andrew Abbie, was established by Mrs. Audrey Abbie for the promotion and advancement of studies in neuroanatomy and in related fields. This evening's special guest lecturer is Dr. James Rutka, who I think likes to be called Jim. Jim is the RS McLaughlin Professor of the Department of Surgery at the University of Toronto. Educated at Princeton University and Queen's University Medical School, Dr. Rutka was an intern at McGill University before entering the University of Toronto Neurosurgery training program in 1982. He took up his appointment in the Division of Neurosurgery, the Department of Surgery at the University of Toronto in 1990. He is a pediatric neurosurgeon whose clinical practice has been at the Hospital for Sick Children in Toronto. His primary research and clinical interests relate to the science and surgery of human brain tumours and epilepsy, and he has over 500 peer-reviewed publications to his name. He served as the president of the American Association of Neurological Surgeons in 2010, and in 2011 became president of the World Academy of Neurological Surgery, president of the American Academy of Neurological Surgery, and fellow of the Royal Society of Canada. In 2015, he received the Margolese National Brain Disorders Prize from the University of British Columbia, the Robert L. Noble Award from the Canadian Cancer Society, and was made an Officer of the Order of Canada. So to discuss human brain tumours, recent advances from the leading edge and beyond, I'm delighted to welcome Dr. James Rutka.
Test. I think it's working fine. Thanks, Alistair, very much for that kind introduction. And when I first received the correspondence to come and give this lecture on behalf of Professor Abbie and this memorial lectureship, there was a lot of back and forth about giving a lecture in neuroanatomy, so I was kind of worried for a while I'd have to dust off my neuroanatomy textbooks and bring that back to the surface. It wouldn't be a problem, however, because I actually enjoy neuroanatomy a lot, and that was probably the main reason why I decided to go into neurosurgery. So I'm delighted to be here, and I'm glad you have flexibility in this lectureship to be able to talk about things outside of neuroanatomy, and I'll be talking about some of the research work that we've done and how that's leading to clinical trials in human brain tumor research. So I'm deeply honored to be here, and Alistair already mentioned a little bit about the person whom we're remembering today, Professor Abbie, and here's some information about him. I know he's very familiar to this university and all the complexes in this university, but truly an amazing academic, and who had studied in the UK, in Australia, was associate dean and then dean of this university, and president of several societies related to anatomy. So I'm really honored to be here, but also very honored that it's a distinguished couple. And Ruth, I think, is the way she'd like to be called. Dr. Abbie's wife was, amongst herself, she was really an incredible force, and she was a gynecologist who, as I understand it, beat back a traditionally very male-dominated field at that time, and had a very vibrant practice of gynecology. I went back to the books and looked a little bit about what Dr. Abbie had published in the past, and here's some of his work that he had published in the Journal of Anatomy in 1950 on the articulations in the skull, also head form and human evolution. And this table that you see, that's kind of the way research was done in those days. And again, this is from the Journal of Anatomy, but just measuring skulls, of different ethnic groups, and then trying to draw inferences or conclusions based on these measurements. And that was the way research was done back in those days. I just want to mention a collaboration that we have between the University of Toronto and the University of Adelaide. You can't see me so well, but that's me against the pillar there at the University of Adelaide. And I really like your jacaranda trees. We don't have those in Toronto. They're really spectacular, especially this time of year. This is from a trip I took here 4 years ago. And I have two colleagues in my field of interest, neurosurgery. So Amal Abu Hamdan and also Steve Santorina. Steve's here, I saw, so many thanks to them. The last time I was here 4 years ago, I went directly off the plane from Toronto to the Barossa Valley, and that was great without stopping. And so it was a wonderful tour and trip that I took with them back at that time. I have no disclosures for this talk relevant to this, but I realize this is a mixed audience. That's what Alistair told me. So I just wanted to mention that as part of the research work that we do in the field of human brain tumors, that sometimes it's performed in animal models of cancer. So I just wanted to make that clear before I started. So for this mixed group, I just want to start with a classification of human brain tumors. And here's an image, sagittal image of the human brain. And these tumors can be classified as supratentorial, that is above the tentorium, which is the membrane that separates the cerebellum from the hemispheres. And these are the tumor types that occur there. And then there are infratentorial tumors, and these are the tumors that can occur in this compartment. What's interesting is that tumors in the supratentorial compartment are largely confined to the adult population, whereas in children you see them more highly represented in the posterior fossa area. And the reasons for that likely relate to the neuroembryogenesis of the cerebellum and how the cells are very rapidly dividing at that time point and how tumors can form at that time. This talk is really a talk about three different tumors. And so one is glioblastoma, GBM. Another one is going to be diffuse intrinsic pontine glioma, or DIPG. And the last bit of the presentation will be about atypical teratoid rhabdoid tumor, or ATRT. So we'll begin first with GBM. This is known as a fatal tumor. Most patients who have this will be surviving about 2 years or less, despite all of our therapies that we have available. It's the most common malignant tumor in adults. It's characterized by this central area of necrosis that you can see here. And then it's got two other features to it. One is a highly cellular proliferative core, which is designated here in the center, but also a rim of invasion going around it. So if you draw concentric circles around the tumor in red here, you'll see that tumor cells will be escaping beyond that central core and will go out into the periphery, and that's one of the hallmark features of GBM. Looking at it on a cellular basis, you can see here the densely cellular core of the tumor and then the cells that escape that core and go into the periphery. And it's characterized on the MRI scans that we do as follows, where you have a T1 weighted sequence with gadolinium. That's contrast that's given to patients that have brain tumors. And it's the central core that lights up with the contrast, where on a flare sequence on the MRI scan, you can see the signal intensity that goes beyond that core, and that's the invading cell population that I'll be talking about. And why patients often fail their treatment strategies is because the tumor cells go far beyond the central core. My laboratory over 30 years now has explored these areas of glioblastoma invasiveness, and you can read from this coronal section schematic of a brain secretion of proteinases, the extracellular matrix of the brain, how tumor cells are very tricky and they themselves can secrete permissive extracellular matrix. They can interact with the extracellular matrix through their cytoskeleton, and all of that can lead to invasiveness. And recently my lab has been devoted to looking at these molecular motors that will drive cancer cells through the brain. So that summarizes almost 30 years of lab experience for you and in various domains that I've been exploring. So we've made some advances, mostly with respect to the understanding of the Rho-GTPase pathway, which I'll talk about in a little bit of detail, and then targeting, like how can we target these brain tumors using a variety of strategies. One I've underlined here is the MR-guided focused ultrasound I'll be talking about in detail, and then functionalized nanoparticles. So I have just a few, maybe 3 or 4 data slides I want to show you, and then I'll spend most of the time more in schematics. But these elements in this pathway, the Rho-GTPase pathway, are very important for cell migration and cellular, and particularly cancer invasion. And the most common ones are the Rho-GTP, RAC-GTP, and CDC42 GTPases. And they're small cytoskeletal GTPases that work fundamentally to change the actin cytoskeleton to allow cells to change shape, but also to allow them to propel themselves into the matrix. The very first person who studied that in my lab was Badur Salia. She's now an associate professor at the University of Southern California. I'm going to show you some data slides on CDC42 GTPases. So where that exists here is in the cytoplasm, and it works downstream here on the cytoskeleton that you can see here. So we'll just talk a little bit about it and some experiments that we did with it. So we had an opportunity to transfect two different glioma cell lines, or GBM cell lines, U87 and U251. And this is the constitutively active form of it, where we had it overexpressed. So a CDC42 GTP is overexpressed here and here that you can see. And the question is, what does that do to the invasiveness and the growth of human brain tumors? And here in this model, you can see that here's the wild type CDC42 expressing cell line that's expressed, and here's the overexpressing CDC42 GTP. And you can see that these animals die faster than these animals once the CDC42 is induced to express with doxycycline. So we published this work a short while ago, but now there are these inhibitors that we can use against molecules in this pathway, and I'll be talking a little bit more about how we go about doing that. Upstream of these small cytoskeletal GTPases, which are found here in the cytoplasm, you have on the cell membrane a number of interactors that come to play. And a lot of these are what are known as the guanine exchange factors. And over many years, my lab has looked at a variety of these. And the red arrows that you see coming into play here are various molecules in this pathway upstream from the cell membrane down to the Rho-GTPases that we've been exploring, and it's been a very fruitful form of investigation. This schematic is showing you the red stop signs, which are drugs or small molecule inhibitors that can work against this pathway, and we've got them all in our possession. The question is, how can we get them delivered to a brain tumor like GBM? So initially, at first, we turned to the whole concept of nanomedicine. This is a paper we published about 8 years ago in New England Journal of Medicine. Warren Chan is a bioengineer working at the University of Toronto, and Betty Kim, I have a picture I'll show you, was a neurosurgery resident at that time, training in my laboratory. But I'll just remind you that the nanoscale is on the order of 1 to 100 nanometers. So that's down here at the bottom of the scale. Here's a baseball. That's 10 to the 8th, and this is now 10 to the 2nd in terms of the nanoscale here. And you can see where red blood cells fit. And we know now that nanomedicine is being used not only for contrast-enhanced delivery of agents, but also for cancer treatment. And there's Betty, and she's now a faculty neurosurgeon at Mayo Clinic. Here's Warren Chan, who was part of the New England Journal paper that we put together, but his whole lab is devoted to producing nanoparticles, synthesizing them, and then having them being able to be delivered to cancers, including brain tumors. So at first we wondered whether these nanoparticles could cross the blood-brain barrier. And I'll be speaking a little bit more about the blood-brain barrier in a moment. But we used this in an in vitro model that we had, and we asked the question, did these nanoparticles go across a in vitro blood-brain barrier? The answer was yes, they did. Arnold Etame is a PhD student. He's now at the University of Michigan, but he got his PhD in my laboratory. So that was in vitro study, but what about in vivo? Can we actually deliver these nanoparticles across the blood-brain barrier? In order to do that, we decided to use an approach with MR-guided focused ultrasound. In this particular approach, you can see the nanoparticle conjugates are these small circles in white, and the blue represents microbubbles that are injected just prior to the delivery of the nanoparticles. And we were very fortunate that we have a maven in Toronto for MR-guided focused ultrasound. His name is Dr. Kalervo Hininen. He's probably the lead expert in the world with MR-guided focused ultrasound to help us with our studies. So to do these types of images, this is the setup you can see here on the left-hand side, where the transducer amplifier system gives us these very in-depth images. Now, these are MR images of a rodent's brain, and you can see the breakdown of the blood-brain barrier that's shown here, where the arrows are pointing. And not only that, but when we see where the gold is deposited after we're delivering it in these model systems, you can see this is the GBM, the tumor down here on the left-hand side, and up above is the normal brain. And right at the invading edge where we want to be is where we're able to deposit the nanoparticles. So we showed that we were able to do that using MR-guided focused ultrasound in the publication in Nanomedicine, and then in neurosurgical focus, we showed that this is kind of a new approach to how to break down the blood-brain barrier and get things across that normally the blood-brain barrier wouldn't allow to cross. Here's some other images. This is without focused ultrasound, and this is with focused ultrasound. You can see the greater rim of enhancement around it, but more importantly, the deposition of the gold nanoparticles that appear black here next to the tumor, but this is normal brain here, and also in this image here. So delivering it to the tumor margins is possible, but now we can also use the properties of gold as a Raman spectroscopy visualizer, and we're able now to coincide the image that we see from the in vivo histological sections right onto the MRI scan that is being shown here, and showing how the gold nanoparticles that are found on a certain region of the brain can be related back to the imaging that we had prior to doing these studies. This is work that we published also in Nanomedicine. And Roberto Diaz, now he's a faculty neurosurgeon at McGill University, but he did his PhD training in my laboratory. So I talked a little bit about going to the invasive rim around the core, but now we're just going to talk a little bit about going to the actual core itself and how to treat glioblastoma with gold nanoparticles that are conjugated to a very common chemotherapeutic used in cancer, which is cisplatin. So here's cisplatin. You can see it here in the upper left. Here's gold and cisplatin together. And we also had cisplatin conjugated to an uptake peptide that allowed for greater endocytosis than would be achieved with the other two methods. And just a proof of principle experiment here showing that in the presence of focused ultrasound, we get far greater concentration of cisplatin into the brain itself on mass spectrometry than otherwise. And this is a model done in mice where we're able to look at, using luminescence and a bioluminometer, the size of tumors, because photons are produced in the method that we use to look at these tumors. And in the presence of gold nanoparticles and cisplatin, you can see the tumors are much smaller. And so this is work that we published just recently this year in Nanomedicine, showing the use of cisplatin and gold nanoparticles, MR-guided focused ultrasound, to treat the core of glioblastoma. So I have a video I'm going to show you. I just want to set the background to this video. I had a master's student who worked in my laboratory who was doing a biomedical illustration master's, and she spent the whole year doing this sketch of a story on focused ultrasound to be used with nanoparticles. So she spent the whole year as one person doing this whole animation and putting it all together. I'm just going to advance it to a point where the story begins, and then we'll watch it. It's about 3 minutes long, and you can see this technique of MR-guided focused ultrasound and crossing the blood-brain barrier in more detail.
Every day, 27 Canadians are diagnosed with a brain tumor. The most common type of primary malignant brain tumor is glioblastoma multiforme. Average survival, even with aggressive treatment, is less than 1 year. One of the main obstacles in the treatment of brain tumors is the effective delivery of therapeutics across the blood-brain barrier. This barrier serves to protect the brain from harmful toxins and chemicals by allowing only certain molecules to leave the blood and enter the brain tissue. So how do we get anti-cancer therapeutics past the blood-brain barrier? One approach is to use gold nanoparticles as carriers. These nanoparticles are able to cross the blood-brain barrier due to their minute size. Once in the brain tissue, tumor-specific antibodies help to specifically target cancer cells. But how do we focus the nanoparticles to the area of the brain that has the tumor? The Peter Gilgan Center for Research and Learning is home to the Arthur and Sonia Labatt Brain Tumor Research Center. Researchers here have found a way to use MRI-guided focused ultrasound to outsmart the blood-brain barrier. This technique is being developed using mouse models of brain tumors. The mouse shown here has a growing brain tumor and can be used to evaluate the delivery of therapeutic-laden gold nanoparticles to tumor tissue. Via the tail vein, the mouse is intravenously administered ultrasound-responsive microbubbles. As the microbubbles reach the brain and the tumor capillary bed, magnetic resonance imaging is used to focus ultrasound waves on the specific area of the brain where the tumor is located. The ultrasound waves trigger transient size enhancement in the microbubbles. The swelling of the microbubbles within the ultrasound field causes microscopic physical disruptions of the capillary lining and results in a localized temporary breach of the blood-brain barrier. The mouse is then injected with the therapeutic-laden antibody-coated gold nanoparticles. The nanoparticles travel in the blood, reaching the area of the brain tumor. The holes in the capillary linings, temporarily created by the microbubbles, allow the nanoparticles to escape the blood-brain barrier and enter into the extracellular space of the tumor tissue. This results in a concentration of gold nanoparticles in the region of the tumor. Tumor cells have receptors expressed on their surface. The antibodies on the surface of the nanoparticles allow them to bind to certain receptors that are enriched on the surface of tumor cells. Binding to the cell surface receptor triggers internalization of the nanoparticle. The internalized nanoparticles release their anti-cancer drugs within the tumor cell, triggering the cell to die. This process occurs simultaneously throughout the tumor, resulting in widespread tumor cell death. This not only significantly reduces the size of the brain tumor, but importantly, eliminates aggressive, invasive cells along the tumor border. With success of this tumor targeting strategy in mouse models, it is our hope to apply this technology to the treatment and elimination of various types of human brain tumors.
Yeah, so obviously she passed and she got her master's. That was pretty amazing animation. She said the hardest thing is, Stacy is her name, and she said the hardest thing about doing the animation was the fur on the mouse. That was the hardest thing. So including for the first tumor I'll be talking about today, I talked about treating the glioma core, so the central region, but also getting at the zone of infiltration around it, specifically targeting the Rho-GTPase pathway. These cut across all types of GBM subtypes, and it deals also with the genotypic and phenotypic heterogeneity, which is characteristic of GBM. And novel treatment strategies with the gold nanoparticle conjugates I mentioned, and using the MR-guided focused ultrasound strategy for going across the blood-brain barrier. So this is now in clinical trials using MR-guided focused ultrasound. We have a trial at one of our hospitals in Toronto, and we're hoping then that we'll be able to use in the near future these novel agents I mentioned to you in some of these clinical trials, and looking at ways that we can trace the gold nanoparticles by imaging. So if you can see where the drugs and reagents are going without having, obviously, to sample the tissue itself, that would be a huge benefit, I think, for this whole field. Okay, so the next tumor I'll talk about is diffuse intrinsic pontine glioma. It's a fatal tumor. Sadly, it's a tumor that occurs in children typically less than 6 years of age, so usually between ages 4 and 8. There you can see what it looks like on a sagittal MRI scan right in the pons. It fills the whole pons, and we have no good treatment for this. In the last 30 years, there's been no benefit to any of the underlying treatments that we've used, including new treatments that have been tried. So we know that the blood-brain barrier is quite intact, however, and it doesn't allow for passage of reagents. And so the question is, can we use MR-guided focused ultrasound to try to get at this tumor in using novel reagents as well? Well, just a few years ago, there were 4 big papers that were published in Nature Genetics all at the same time. The top one is from a French group. This is from the group at St. Jude's in Memphis, and then this is a Boston group, and then this is a group from our hospital. Cynthia Hawkins here published this, and you can see one of the new genes that came out of an analysis of DIPG was the ACVR1 gene. I'll talk about that in a moment, but this was published in Nature Genetics in 2014. I'm going to show you a case of a 16-year-old male who had symptoms and signs suggestive of a DIPG. He was otherwise fit and had no past medical history of note. So you can see there it is occupying the entire pons on this axial MRI scan. Although the age isn't typical, we decided not to do a biopsy. He went on to get radiation and chemotherapy, and sadly he passed after only a year and a half. And this is his MRI scan on treatment. So his name happened to be Daniel, and you'll see why that's important, because he and his family have done a lot of work on raising funds for efforts. And this is a news piece that came out when the gene was discovered for DIPG, some of the genes that I mentioned to you. And this is a news piece from Toronto.
New research is out on a deadly form of brain cancer. DIPG affects children and teens, and over 90% of those afflicted die within 18 months of being diagnosed. But as Stephen D'Souza explains, researchers in Toronto say they found a genetic clue about the disease.
This is on our trip to Dominican when we went down after we had just gotten the diagnosis.
Stan and Marisa Bertoya haven't changed their son Daniel's room since his death 6 years ago. At age 16, Daniel was diagnosed with a rare brain tumor and was told he had less than a year to live.
Losing your eyesight, losing your hearing, losing your speech, losing your mobility, basically just devastating to see your son going through that.
His parents scoured the world for help, but found no effective treatment.
We hung on, to every little thing that we could possibly hang on to, but really there was nothing there.
Before his death at age 18, family and friends organized fundraisers, and Daniel had one wish for the money.
He said, "Dad, I want it directly to research to help find a cure."
And this big white blob in the middle is the tumor.
Daniel's type of cancer, known as DIPG, is diagnosed in a few hundred young people in North America each year. Located on the brainstem, it's inoperable and chemotherapy is ineffective. But using new technology, researchers at the Hospital for Sick Children discovered cancer genes they hadn't seen before.
The genes mutated in this cancer are completely different than what we see in adults, and so we cannot use the same drugs as what we were using in adults.
That told them they needed new and unique treatments.
We can go to somebody with this diagnosis, and rather than it being a death sentence, maybe there's some hope that some chemotherapies are going to work now.
While that's still years away, after raising more than $500,000 for research, Marisa and Stan are happy to see progress.
To get this little baby step right now, it's just amazing, and I'm hoping that, it'll turn into giant steps one day.
His favorite player.
They say this gives them even more motivation to keep raising money and awareness to help fulfill their son's final wish. Stephen D'Souza, CBC News, Vaughan, Ontario.
Okay, so this is a little bit about the ACVR1 gene that was discovered recently for DIPG. Here it is on the cell surface, you can see, and then it signals downstream through the SMAD transcription transactivators, leading to cell cycle changes. And now it's becoming a good target to think about for DIPG treatment, and a number of model systems have come forward looking at that. What we decided, however, to do was to see if we could get reagents to the pons, into the brainstem, using MR-guided focused ultrasound. And we had an experimental approach where we could target the pons in mice, and then see if we could break down the blood-brain barrier and achieve higher concentrations of reagents there than you normally could do without this technique. We showed it was safe to do physiological monitoring. There was no difference in heart rate, blood pressure, respiratory rate of these animals who were undergoing focused ultrasound treatment. Here are some histological images taken after we used focused ultrasound. Evans blue dye was used because it will cross the blood-brain barrier if it's broken down. Here you can see just with microbubbles by itself, here just with focused ultrasound by itself, and now this is focused ultrasound and microbubbles. You can see the blue staining within the pons. Some of it also gets into the cerebellum, but we get high concentrations where the rest of the brain is not affected. So it was a very useful strategy to try to achieve higher concentrations of reagents. And this is the histology shown at various sections of the brainstem. There's no vacuolization. There's no change in the actual tissue itself from the brainstem. And using various stains for apoptosis, there was no evidence of apoptosis happening as well. We then tried to use a reagent to cross the blood-brain barrier using this strategy. These are 3 different DIPG cell lines that you're seeing here. It's a heat map for drug sensitivity. Red means higher sensitivity. And the one that had the best sensitivity was doxorubicin. And that's a good drug to try because it doesn't cross the blood-brain barrier, at least not readily. So we decided to use doxorubicin and see if it would cross the blood-brain barrier using focused ultrasound. And in fact, we can get almost 400 times the concentration of doxorubicin using focused ultrasound than what we could do without it. So this is work that we just published this year in the Journal of Controlled Release, and showing not only its safety, but hopefully its efficacy. And with this preclinical work to show that this can be achieved, and with a disease that has literally no more than 18 months of survival benefit, we've moved to writing up a clinical trial to try to use this in children with DIPG, and we launched this last year. It has gone through Research Ethics Board approval, and we're about to enter our first patient coming next month. So we're hoping that this will lead to some better reagents going across the blood-brain barrier, better opportunities than to treat DIPG, and also thinking about the genetics and some of the new targets that are out there, being able to use the 2 techniques combined to have a better outcome for children with this very, very bad tumor. Okay, so the last tumor then that I'll talk about during this presentation is the atypical teratoid rhabdoid tumor, or abbreviated to ATRT. And when I was a junior staff man at the Hospital for Sick Children in neurosurgery, we came across a young child, 2 years old, who had this tumor that was called a malignant rhabdoid tumor. It was in the pineal region, and there it is there, and there you can see after surgery, and this child died rather rapidly after the surgery and after chemotherapy was given, despite our best efforts. And so we've come a long way with our understanding of this tumor since that time. It was first described in 1987. It was called malignant rhabdoid tumor, but we now know that that refers mostly to the kidney tumors that occur. But in 1996, they were reclassified in the brain as atypical teratoid rhabdoid tumor, or ATRTs. They can occur in a variety of locations: the supratentorial compartment, the posterior fossa, also the spine. They have one hallmark feature genetically, and that is loss of a gene that's called SNF5. And the diagnostic feature for this is negative staining by immunohistochemistry for the protein product. It's also called BAF47. So these are some of the background pieces that you need to know for ATRT. Our group, especially Annie Huang at our institution, has done a lot of work doing a molecular subclassification of ATRTs, and it can now be classified into Group 1, Group 2, and Group 2 is further classified into 2A and 2B. The Group 1 is sometimes in the supratentorial, sometimes in the posterior fossa. Group 2A is exclusively confined to the posterior fossa, and Group 2B can occur across the neuraxis that you can see here. And the subclassification of ATRTs was described by Annie and her group in Cancer Cell about 2 years ago. So Brian Goldburn is a PhD student working in my laboratory, and he wanted to model ATRT because we never had a good in vivo model to try to study how we could target this particular tumor with better reagents. And so Brian thought that if we targeted a primitive, let me just go back, if we targeted a precursor cell, like a neurogenic precursor cell, and they're delineated by this lineage marker that's known as brain lipid binding protein, or BLBP. If you could target that and somehow knock out the SNF5 gene at that early stage in neurogenesis, then maybe you could get a genetically engineered mouse that would have a tumor very similar to ATRT. So we did that strategy. We used the Cre recombinase system that allows you to flank the SNF5 gene and then to knock it out using the Cre recombinase system. And when we did that, we got this mouse at the bottom here. And interestingly, there was a shortened survival that you could see here compared to the wild type, where the SNF5 gene is still expressed. And we thought, okay, maybe we have now generated a mouse with tumors. It turns out here's the mouse on the left. This is the knockout SNF5 gene mouse here. Here's the wild type here. You can see they have different sizes. They look different. On the other hand, the cerebellum is completely disrupted in the SNF5 knockout mouse that you can see here compared to the normal control mouse that is here. But unfortunately, none of the mice developed brain tumors. So although we had a good strategy, unfortunately, we didn't develop a system whereby tumors were found. So we looked more closely at the histology of the genetic knockout in BLPP precursor cells, and although there were some of these dense cell masses that we're seeing here throughout the brain, these masses tended to disappear over time. They underwent apoptosis and they didn't form tumors. So we went back to the drawing board and we thought about revising our hypothesis to come up with a strategy to knock out a precursor cell or a stem cell earlier in neuroembryogenesis to see if that would lead to a mouse that would develop a tumor like ATRT. So we targeted the knockout of an SNF5 gene by day 6.5. And the way we did that was with an inducible knockout system using the Rosa-Cree system here. And this refers to the estrogen receptor that is turned on by tamoxifen administration. So when you give tamoxifen at 6 days after conception, you can theoretically knock out the SNF5 gene. And that's what we did with this breeding strategy that you can see here. We ended up with a mouse that had this Kaplan-Meier survival that you see here. It looks similar to the previous one. And again, this is when it was turned on with tamoxifen. Question is, did these mice then develop brain tumors? So the answer is yes. So we developed finally, after much experimentation, a genetically engineered mouse model now where the mice develop brain tumors that look like ATRT, specifically in the hindbrain, but all throughout the central nervous system that you can see. And they have highly proliferative indices and the same spatial and patterning that you would see in ATRTs. We showed that these ATRTs, in the hindbrain in particular, had the worst prognosis, as was shown here, compared to other elements of the central nervous system. And we showed that these tumors were highly proliferative here on the Ki67 staining that you can see on the immunohistochemistry shown on the right-hand side. And in terms of survival, you can see, or at least this is the proliferation Ki67 indices, you can see the highly proliferative, especially the hindbrain component as is shown here. So by modeling now an ATRT that gives us a lot of opportunity, we were able to show that we could target early in embryogenesis, a time point, knocking out this gene that's critically important to forming ATRTs, and then having a model now that allows us to test ATRT using novel reagents. This system proved to be very valuable in terms of knockout. So the question is, do these particular mouse models, do they represent what happens actually in the human models or in humans? And then can we try to treat these models in ATRT in mice with targeted therapies? So then we'd like, obviously, to bring that into clinical trials. So we actually have a high throughput drug screening strategy that we utilized. And at first we looked at targeting kinases. These are enzymes that are very important in signal transduction pathways, and they help to expedite these signaling pathways. And so we had a library of 477 of them, and we looked across group 2 and group 1 tumors for ATRTs and looking at their sensitivities to the various kinase inhibitors. We had about 11 that were found in the group 2 group, and then about 20 in group 1 that you can see here. One in particular that was very interesting was the ERB2 that I'll show you more data on. So in group 2 tumors, you can see that ERB2 is expressed, but it's not expressed in group 1. These are different cell lines for ATRT in vitro. And then when we knocked out ERB2, as shown here using a siRNA strategy, which basically eliminates small molecule elimination of ERB2, we could also see that phospho-AKT is also involved, and so it seems to be involved in signaling through the PI3 kinase and the AKT phosphorylation signaling pathway. We then looked at doing radiosensitization studies and drug synergy, and for radiosensitization we used sublethal doses of radiation combined with drugs themselves, trying to see how the combination of drug and radiation worked in ATRT cell lines. And we found that for the group 2A, 2B tumors, these were the various pathways that were looking interesting. Here's the ERB2 again, but also BCR-ABL was of interest. In terms of broad-acting subset of pathways, PI3 kinase, mTOR pathway, MAP kinase, CDK2, and so on, all of these look very interesting for treating ATRTs now in the future. We then looked at combining drugs, so using combinations of drugs, how do they appear, and it looks like a subtype-specific drug that looks very exciting is dasatinib for the group 2 tumors, and then more broad-acting tumors like rapamycin, dramatinib, and also dinaciclib that's also used for testing in and against ATRT. So what we've shown from these studies in ATRT is that group 2 tumors, this is the drug testing side, are sensitive towards these multi-kinase inhibitors. We've also been able to show that ranking inhibitors for broad-acting activity by potency reveals that these cell lines are sensitive to these two pathways, PI3 kinase, mTOR, and also RAS MAP kinase signaling pathway. And that combination studies of drugs shows dasatinib looks very interesting now for ATRT. It hasn't been utilized before to any significant degree and has the capacity to radiosensitize, so combination of radiation therapy and drug therapy together acts synergistically and probably along the lines of inhibiting these two pathways and their signaling. So our future directions is to evaluate these inhibitors in combinations using these ATRT as xenografts in vivo and also in patient-derived xenografts. But we'd also like to test, obviously, our genetically engineered mouse model that we've created to be able to do that. Brian Goldburn, the fellow who did the PhD studies for the ATRT work and knocking out SNF5 at a critical time point that led to ATRT tumors, is shown in the middle here on the day of his defense just this September, and so he's now graduated, and we're delighted for the success. That was 4 years of work to create a mouse model, just so you get some time appreciation of how long it takes to build some of these models. So it took 4 years for him to build that model, and now we're using it hopefully to find new drugs that will be available to treat children with ATRT in the future. So in conclusion, I told you about 3 tumors. One was GBM and the possibilities of gold nanoparticle conjugates and MR-guided focused ultrasound, and how that's leading now towards clinical trials, which is becoming very interesting and exciting. DIPG, enhanced delivery of doxorubicin, looks very promising in the ponds, and we're launching a clinical trial to test that hopefully within this next year. Some results will be available. And then I told you about ATRT and the new mouse model that we developed and drug sensitivity platforms that we used to find drugs like dasatinib that look very interesting, targeting the PI3 kinase, mTOR pathway, and the like for hopefully leading towards more preclinical studies. This is our research team in Toronto. So we have probably the largest brain tumor research center, at least in North America, perhaps the world, that is studying. This is over 100 individuals that are studying the problem of brain tumors. It's called the Arthur and Sonia Labatt Brain Tumor Research Center. Arthur and Sonia Labatt are famed philanthropists who gave significant funds to help us kickstart our project more than 10 years ago. And now over 117 researchers are working on the problem of brain tumors of a variety of sorts. We're in a children's hospital, so we are studying a lot of the pediatric brain tumors, but also adult brain tumors are being studied in this research-rich environment. This is my own personal laboratory, and what I'd like to just point out are the individuals here on the right-hand side who did a lot of the work that I mentioned today. Special thanks specifically to Badur Salia, who got the work going on the Rho-GTPase pathway in my laboratory. Betty Kim did the work on nanoparticles with Warren Chan, collateral Hinanen, MR-guided focused ultrasound. When we looked at blood-brain barrier and crossing, Arnold Aitami did the first work of an in vitro blood-brain barrier. Daniel Caluccia and Carlin Figueredo were important in the recent paper that we published on gold cisplatinum nanoparticles against GBM. And Brian Goldburn did the studies, as I mentioned, looking at ATRT and developing the genetically engineered mouse model of ATRT. And so here's the lab group. Here's some of the funding sources that I've been very privileged to have over the years. And with that, I'd like to conclude and just thank all of you. And I have a deep appreciation and thanks for having been given this opportunity to give the Abbie lecture here at the University of Adelaide. I thank you for your attention.
Thank you very much, Jim. Questions from the audience? Yes, please.
Is the biology of the tumour infiltrating astrocyte and the diffusely infiltrating astrocyte different from one with a mass?
Yeah, so the question is whether you start first, let's say, with a diffusely infiltrating astrocytoma, where there's no single mass, but rather the whole substrate of the tumor is infiltrating, versus a tumor like I demonstrated, which is GBM, which has a central mass, and then beyond it, a rim of invading cells. I think the answer to that is, yeah, they're genetically different. They have different genetic markers, and to the extent that the rim cells around the core have been studied in GBM versus a diffuse astrocytoma, those infiltrating cells have been shown to have different biological but also molecular properties.
Other questions? Perhaps I could ask about the challenges of clinical trials in some of these tumors, because these are relatively uncommon malignancies. So the DIPG, the ATRTs, what sort of numbers do you require to have power to make those clinical trials work?
Yeah, so ideally it would be great if you could get, the question is about clinical trials, if you could get multi-centered trials and accumulate lots of patients across the country. Yep. Canada has a small population, but if you pooled all the patients with DIPG across the country, probably in the course of a year you'd have maybe 25 patients, so that's still a very small number. Yep. If you added the United States and North America, then you could, 10 times the number. So we're launching our DIPG trial as a pilot study out of Toronto with small numbers, looking first at safety and efficacy, so small numbers. That's what we would hope to prove, that it is not only safe, but possibly maybe have some efficacy. And then we would launch into, we would hope, a multi-center trial across North America. In terms of other trials that are being done now in North America, most of them are done through the cooperative trial strategy, where you have numerous centers interacting with and collaborating with an idea and putting that into practice with a clinical trial that's been thought out very carefully and has probably some pilot data already, and then is leading towards a more practical approach across many patients across numerous centers. But the challenge is that most of the trials, at least in North America, are pretty boring, meaning that the drugs that are selected, the reagents that are used, are likely known to fail. They just aren't thinking outside the box. But recently, some smaller cooperative trials are starting that look more exciting, because they're willing to take a bit more risk and to try novel reagents that hadn't been tried before. So I think there's room for some optimism that we're not wasting our time, that there actually may be some benefit accrued to the patients who are undergoing these treatments.
Thank you. Yeah.
So the question is, how does let's say you knew something about the molecular strategies or genotype of these tumors before you did surgery, or even if you were doing surgery, let's say, and you wanted to know, is it going to be better to go for a gross total resection, or is it better not to take a risk for something like that and cause harm to the patient, because you know that the molecular genotype is going to allow you to back off and allow some other adjuvant therapy like chemotherapy or radiation to work. So maybe a good example of that would be applications that we know exist for medulloblastoma. So I know it's a pediatric tumor. So medulloblastoma is the most common malignant tumor in childhood. I didn't talk about that today, but from our center, our group has done a lot to demonstrate what the molecular subtypes of medulloblastoma are. And if you knew that your patient had a Wnt type medulloblastoma, there are 4 main subgroups of medulloblastoma. There's Wnt, sonic hedgehog, group 3 and group 4. Those are the main types. But if you knew it was a Wnt subtype of medulloblastoma, you could definitely back out earlier than having to perform a gross total resection, especially if the tumor was abutting the brainstem and you were worried about having, a brainstem insult from doing tumor surgery, because we know that those patients do extremely well with modest therapy afterwards, including chemotherapy, and they don't need to get radiation. So that's one example of how the molecular information, if you had that available at the time of surgery, would influence what you would do. Now, most of the time we don't have the benefit of having molecular information at the time of surgery. However, our group now is collaborating with a scientist at the Toronto General Hospital who is using mass spectroscopy and spectrometry intraoperatively to make genetic diagnoses. And we just published on being able to diagnose different subtypes of medulloblastoma intraoperatively using this strategy, and then you could apply what you know from the genetics to that operation on the patient. Yeah, Steve.
There's currently, I think, a couple of trials in DIPG in Europe, which involve target brachytherapy, a stereotactic insertion of guided catheters into the brainstem and delivering various therapeutic agents. Perhaps if you can comment on that.
Yeah, so the question is about other forms of therapy for DIPG. I mean, and you mentioned putting catheters into the brainstem as a solution. I think the person who's doing the best work, Steve, on that is Mark Swadan. He's out of New York University. And what he's done is he's using convection-enhanced delivery where a catheter is inserted into the brainstem, and they're then infusing chemotherapeutics that will fill a good portion of the brainstem. The problem with that strategy, however, is that most of the time the chemotherapy sits almost in a pool by itself and doesn't infiltrate the pontine substrate itself very well, whereas if you come at it from the vascular side and you, let's say, used MR-guided focused ultrasound, and you can then use the blood vessels themselves to be the conduit of having chemotherapeutics reach the pons, you get a much, we think, better and greater distribution than what you can achieve with convection-enhanced delivery by itself. So while it looks promising, I think Mark Swadan's done some great work, and he's done it very stepwise, and it's been incremental science. It's being shown that in trials that it's not getting exactly the result that we had anticipated, although he's now moving to using some very bold reagents and some immunotherapy strategies that look kind of exciting with convection-enhanced delivery. So we'll see what that holds in the near future.
Yes, Simon.
Yes, so thank you. The question is about MR-guided focused ultrasound. How long can you repeat it, the strategy? Are there any caveats or downsides to using the technique? So I didn't mention, but what happens with the strategy to break down the blood-brain barrier with the microbubbles is that there's a transient disruption of the endothelial cells within blood vessels that lasts about 4 hours, and then it's reconstituted, and the barrier is back intact again. Claire Vilhinen has shown this repeatedly in a number of animal studies. So, because the feared complication or the consequences of MR-guided focused ultrasound is hemorrhage, right? The last thing you want to have are red blood cells traversing the blood-brain barrier causing an intraparenchymal hemorrhage. So, with that in mind, you have 4 hours in the blood-brain barriers back to its integrity. We haven't done repeated doses of chemotherapy in the course of a day in an animal model, but what we've done is done it on alternate days, so we know that we can repeat it safely every 2 days, and the animals do quite well. But one of the goals of our clinical trial will be to try to optimize what the dosing scheme and the rate of application of MR-guided focused ultrasound can be, keeping it safe and yet not having any downstream side effects that would be poor for the patient.
All right. Well, thank you very much. I think we'll draw that to a close. I was just reflecting on your title, how apt it was, recent advances from the leading edge, because I'm sure you'll all agree that you've heard something from someone tonight who really is at the leading edge of his field. Thank you for a marvellous update on malignant brain tumours. You've been a very worthy Abbie Memorial lecturer. Now, as a memento of the Abbie lectureship, we would like to award you with this. If I can see if I can get this out.
Oh, beautiful. Thank you.
This is the sterling silver cup, which was handcrafted in 1981 by the late Mr. John Hale, who was an Adelaide silversmith. And it's called the tumbling cup, in that it can't be knocked over. You see the sort of, and as such, it was said to be very useful at sea for having your wine as you're sailing.
Wonderful.
The case that it's in was also handcrafted by Mr. Hale and is made from cedar wood that was originally obtained from a pew in the now demolished Perry Street Church in Adelaide.
Thank you. Thank you very much.
Thank you once again.
Yeah. Thank you, Alistair. Thanks all. Thank you all for coming.
Yes, and thank from me for all coming. So, good evening.